Hello, and a very warm welcome to today's Bioinsights webinar titled Organ-on-Chip Innovation, Modeling the Complex Tumor Microenvironment for Drug Discovery. I'm Abby Pinchbeck, an editor at Bioinsights, and joining me today are Tom Sommermann, Alina Diepenbrock, and Nicole Teusch, who will explore the modeling of the multicellular tumor microenvironment of pancreatic cancer on a fit-for-purpose vascularized spheroid-on-chip model. After the presentation, we'll have a live Q&A session. So we do invite our audience to pose their questions to Tom, Alina, and Nicole using the Ask a Question box at the bottom of your screen. And we'll get to these in the session today. Now I'd like to introduce our presenters. Tom Sommermann completed his PhD at Harvard, where he worked on cell signaling and cell metabolism of lymphomas associated with Epstein-Barr virus. At Dynamic 42, Tom is now heading up the cancer team and working on development of cancer spheroid-on-chip models. Alina Diepenbrock completed her master's degree in molecular biomedicine with a focus on immuno-oncology, investigating dendritic cell-induced CMV-specific T-cell responses in glioblastoma patients. She is currently a PhD candidate at the Institute of Pharmaceutical Biology and Biotechnology at HHU, focusing on the development of an immunocompetent 3D biochip model of pancreatic ductal adenocarcinoma for preclinical drug testing. Nicole Teusch holds a PhD in cell biology from the Scripps Research Institute in La Jolla, California, and has had several positions in preclinical drug discovery in Big Pharma before pursuing her university academic career. Since 2022, she has worked as Managing Director of the Institute of Pharmaceutical Biology and Biotechnology at HH University in Düsseldorf. So without any further ado, I'll hand things over to Thomas to kick off the presentation today. Hi, Tom. Can I just double check you're not on mute there?
Oh, wonderful. Others again. Hello, also a warm welcome from my side. I'm Tom. I will first give an introduction to the actual model, so to the organ-on-chip model, and then afterwards Alina will mostly talk about the biology. So this was a joint collaboration, as I said, on one side, Heinrich-Heine University in Düsseldorf, with Alina then giving the biology later. From our side, we are Dynamic 42. We are a contract research organization here in the middle of Thuringia in Germany. We mostly do drug screening, tox testing, and a lot of model development, especially when it comes to cancer, which is my forte. We mostly do model development for partners in pharma and biotech. And then, of course, we also sell that organ-chip platform for academic clients so that we also empower them. Also, this being said, the work today was funded by the BMBF as a project funding. So what is actually organ-on-chip? Well, if you ask an AI what an organ-on-chip is, you might get something like this. Certainly, maybe nowadays, AI is a little more clever. This was 2 years ago, but if you ask a scientist, of course, organ-on-chip is something different. So what we are talking about are small devices with multiple chamber and channel systems where you can seed in your cells and then grow organ-like structure within this biochip to ask your more complex questions. So it's a more complex in vitro method. And why do we actually need this? Well, organ-on-chip really tries to position itself somewhere in this triangle between the human biology and the humans, animal testing, and the traditional 2D testing methods. So if you look at animal testing, you get the whole complexity of a body, but really you're lacking the human biology. And especially if we're now going towards new drugs in a way of targeted drugs, especially antibodies, immune modulatory effects. These are very, very different between different animal species. And there, of course, you have to go somewhere closer to the human biology if best really use human biology. On the other hand, you have the 2D testing here on the lower right. So here you have the human biology, but you're really lacking the complexity. It's usually single cells, maybe a little co-culture, but really the complexity of a real 3D tissue is missing. And organ-on-chip really tries to position itself somewhere there in the middle, where you keep the human biology, but also have the complexity and keep also the throughput to high enough so that we can ask meaningful questions. It's not just kind of us as a company and maybe some academic groups saying this is really also being pushed by big agencies like the FDA. So the two modernization acts of 2022 and 2025, FDA is really pushing towards replacing animal experimentation with these more complex 3D models, where organ-on-chip is one of them. And really, especially now, like last April, there was a big announcement that FDA really tries to fade out animal experimentation for some type of drugs. So what does organ-on-chip actually look like in principle? So what we are trying in organ-on-chip research is trying to really recreate the minimal functional unit of an organ. So we're trying to zoom in, for example, here, zooming into the gut and really finding the small, usually interactive surfaces of different cell types, where really then the magic of this given tissue is happening. For example, in the gut, this interactive surface between the blood vessel, the endothelium of the blood vessel, and then on the other side, the epithelium, so the gut where the nutrition is taken up. Usually this is arranged around the barrier tissue. This is why you're using these porous membranes that you can see there in the middle. So you would have on the one side endothelial cells and the other side the epithelial cells creating this barrier tissue. You can, for example, do the gut, as I'm showing here, but also, of course, lung would be a wonderful model for the air and blood barrier there, or the blood-brain barrier between the brain and the blood circulation. The cool thing about organ-on-chip is you're not having just this complexity by adding multiple cell types. So this is something you can see directly, right? You're adding endothelial cells and epithelial cells together, but you can additionally get complexity through stress that you're imposing onto the system. So firsthand, you impose shear stress. So usually these chambers are perfused. So the endothelium gets perfusion with a blood media, which then really kind of stresses these endothelial cells, so they start behaving more closely to the real endothelial cells that, of course, also have this shear stress with the blood passing by. And on the other side, for example, you can also perfuse then the gut cells, so gut epithelial cells. So here you would mimic the motion, peristaltic motion in the gut. Besides the shear stress, you can also have stretch and strain, so that membrane in there is slightly flexible. So every time you perfuse, which you're doing in a peristaltic way, you're stretching and straining your tissue, and this also adds kind of to the complexity of the system. Furthermore, you're adding oxygen and nutrition gradients. Oxygen and nutrition is coming in through the vasculature, how it would also look usually in the normal body, and then you're creating this oxygen or nutrition gradient inside of your chip, which will also change how the cells are behaving. A very tiny example for this is really then looking at the gut. So here we're looking at a histology of the gut. Maybe we can look at the very left. You can see like a cross section through the membrane. So the middle gray thing is the membrane, and you have the very thin endothelial cells on the bottom, and then this epithelial setting on the top. And this is how you look when you culture that static. And if you start perfusing this, you can see how the gut epithelial cells are starting to suddenly create this villus-like structure. So really here you see that the dynamic matters dramatically. So this is why organ-on-chip is really cool, and you can make very cool models with this. Now how the Dynamic 42, so our company, is approaching this. I'm going to show you how our organ-on-chip platform actually looks like. What we are working with is this biochip here. So it comes in microscope slide format here, like the hand to really see how big that is. Always comes with two independent experiments on one chip, and then two different, basically chamber systems. And if you look then, zoom inside of one of these chambers, you can see that you have multiple channels, all separated by this porous membrane in the middle. And then you can see different cell types on the different membranes or surfaces. Importantly, then you start also the perfusion. Perfusion is independent, two sides, so you can have one media perfusing like an endothelial media on the top at high speed, and then maybe an epithelial medium on the bottom at low speed to really mimic this blood flow, and on the other side, the gut motion. The biochip itself comes in a few different sizes, depending a little on how much material you have. It's generally made to be compatible with drug testing. So all the material was tested to be compatible, of course, with cell culture, but also on the other hand, compatible with minimal drug absorption so that you can do drug administration studies in there. Once you have the chip, you really don't need much of accessories around this. You need tubing, plugs, and reservoirs to really put this together, what you're seeing on the right. But this is all supplied with the biochip. And then you really need normal tissue culture equipment, so pipettes, a peristaltic pump, which you can also get from us, and then an incubator. And then you put this into the incubator, and you're ready to go with your experimentation. Generally at Dynamic 42, we've set up multiple models. Most of the drug screening and tox testing is done in liver, intestine, lung, and cancer models. But others have already done multiple other entities of organs already in our system. So generally, the possibilities in there, of course, every barrier tissue of the body can be basically mimicked in an organ-on-chip assay. What's very important for us are the different readout options our chip is giving us. We're having about a 48 wells in the normal chip size. We're having about a 48 well size of tissue that you have there. And then you really have the opportunity to always sample the supernatant. So you can always look for secreted proteins. You can always recover the tissue in the end and then look in IF staining or recover RNA, recover protein to look, do your normal biochemistry that you're used to. Also, because they're microscope slide format and have this little imaging window on the side, you can always look live and, for example, track immunofluorescent or fluorescent labeled PBMCs as they're moving through the chip, invading into the tissue. So there's always the possibility to always look live. And of course, you can look for enzymatic activity, transport activity, as you always have like an upper and a lower channel where things are then transported. So this is the general chip, and then on the last minute, I'll take to introduce the chip that was actually used in the project of Alina. So here we're talking about a 3-channel chip, so slightly more complex than what I've shown before. Again, we have this chip with the 2 chamber systems. And if we're now zooming into that right chamber of that chip, you can not see 2 channels, but 3 channels. Again, the upper channel separated by the incubator with a bonding foil, and then we have this membrane here with separating the first from the second channel, where then an endothelium could be sitting on. A second membrane with microcavities in there. So also this is porous, but there are additionally small cavities in here where spheroids can be seated in. They have 800 micrometer in diameter, so we can use then up to 25 spheroids in there, and then a bottom channel that can be used also then to sample spheroid-conditioned media. And with this, I set the stage for Alina, and then she can show what cool biology we can do in this.
Yeah, thank you, Tom, for the nice introduction of the model we used, and I will go on and present the key results we recently published in our paper calling Modeling the Multicellular Tumor Microenvironment of Pancreatic Ductal Adenocarcinoma on a Fit-for-Purpose Biochip for Preclinical Drug Discovery. This was published this year on Lab-on-Chip using the dynamic biochip. So why we choose to work on pancreatic ductal adenocarcinoma, or short PDAC? This cancer has a really low 5-year survival rate with only 13%. This is mainly due to the fact that over 80% of the patients already have an advanced state of disease at the time of diagnosis, making the treatment options very limited. This is also due to the fact that we have a lot of clinical failure of new drug candidates, limiting the treatment options we have available for patients at the moment. Why we have clinical failures, this is mainly reasoned in 2 parts. On the one hand, we have the lack of multicellular human-based in vitro models to get a good translational model for preclinical drug testing. And also this is due to the fact that we have a really complex tumor microenvironment. This is on the one hand really highly fibrotic and desmoplastic, and on the other hand immunosuppressive. Getting a little bit deeper into the tumor microenvironment, you can see here that we have a composition of various cell types. So on the one hand, we have the tumor cells in the center that are surrounded by a large amount of fibroblasts. These fibroblasts produce extracellular matrix proteins. These proteins and the fibroblasts together form a really desmoplastic tumor microenvironment and form a physical barrier around the tumor cells, shielding them from chemotherapeutics as well as from immune cells. Coming to the immune cells, we also have various immune cells present in the tumor microenvironment of PDAC, but they are mainly immunosuppressive tumor immune cells, for example, M2 macrophages or regulatory T cells. So our goal was to model this complex system in an on-chip platform. Therefore, we used the biochip from Dynamic 42 that has the 3 channels. And the bottom channel we used for perfusion with medium. In the middle channel, as Tom nicely explained, we have these microcavities. In these microcavities, we seeded PDX spheroids. These spheroids are composed of tumor cells as well as of fibroblasts, modeling the tumor microenvironment. And in the top channel, we have an endothelial layer over which we can perfuse immune cells as well as drugs. So this is the setup, how it looks. And now I'm coming to the results and how the biology looks. So in A, you can see how the spheroids fit into the microcavities. We have two different types of spheroids we use in our lab. The co-culture spheroids are composed of fibroblasts and tumor cells, while the PANC1 spheroids are just composed of tumor cells. You can see that both types of spheroids nicely fit into the microcavities, and that the co-culture spheroids, as you can see in B, are composed of the fibroblasts and the tumor cells, and the fibroblasts form a shield around the tumor cells in the center, mimicking the situation we find in vivo. As you can see in C, we are able to achieve a good viability when cultivating underflow on the chip, proving that this chip model is suitable for applying our spheroids. The next step is was to establish an endothelial layer in our chip model. As you can see via IF images, we were able to establish a nice endothelial layer. This was intact also in the presence of our co-culture spheroids. As you can see on the right side, we had a increased leakage into the middle channel when we applied our spheroids. That was reversed when we added matrigel, or other component of the tumor microenvironment as an extracellular matrix. So then we went on and used this model for first proof of concept for drug administration. Therefore, we choose the FDA-approved drug vorinostat, also known as SAHA, and we applied it in increasing concentrations over the flow on our chip model. As you can see via IF again, the endothelial layer maintained intact with increasing concentrations of SAHA up to 3.5 micromolar. However, we see a morphological change in our UVEC cells due to the application of a drug. And on the right-hand side, you can see that we get a nice dose-dependent reduction of the cell viability in our spheroid model, really proving the fact that this model is suitable for applying drugs and getting dose-dependent effects on the spheroids while maintaining an intact endothelial layer. The next step was to introduce immune cells into this model. Therefore, we choose monocytes to start with. We isolated monocytes from blood of healthy donors and then perfused it over the endothelial layer for 72 hours. As you can see in A, we had a nice infiltration of the monocytes into our spheroid model, indicated by the CD45 and CD163 positive signal. As we know from the patients, the monocytes differentiate into macrophages, and in particular into M2 macrophages, the immunosuppressive phenotype of macrophages. And to elucidate if our model can reproduce this effect, we did a FACS analysis of the infiltrated monocytes and took a look at the M2 marker, CD163 and CD206, as well as the M1 marker, CD86 and HLDR. And as you can see nicely, we get a nice upregulation of the M2 markers, while we have a significant downregulation of the M1 markers, indicating that the monocytes that infiltrate into our spheroid model then differentiate into macrophages and then further polarize into M2 macrophages, mimicking the situation we find in the patients. So to get a more deeper understanding of our infiltrated macrophages, we then took a look how they localize within our spheroid model. Therefore, we did a whole Z-stack of our spheroid after perfusion on the chip and stained with the macrophages in green and the fibroblasts in blue and the tumor cells in purple. And then did a 3D image of this whole image spheroid and cut it in half. As you can see on the right, So we have the blue shell of the fibroblast, the core of the tumor cells in purple, and indicated with the white arrows is the localization of our infiltrated macrophages. As you can see, the macrophages only localize in the fibrotic part of our spheroid model. This is mimicking the localization we also find in vivo in our patients. So taking this to the next step, we have, of course, a lot of room to improve this model and to further elucidate the effects we see here. And therefore, I want to hand over the word to Nicole to give you a little insight on future perspectives.
Yeah, thank you, Alina and Tom, for this nice presentation. And I would just like to add a few words to this. So despite its simplicity, our model recapitulates key components of the tumor microenvironment of pancreatic adenocarcinoma. However, distinct cell types relevant in the tumor microenvironment are not yet reflected, obviously, in our model, such as, for instance, T cells, who might also be highly relevant also in terms of developing novel therapies. So prospectively, we plan to implement patient-derived organoids, for instance. For instance, this will enable more personalized and translational models, and it will probably allow to identify novel therapies, which are then, let's say, precision-based strategies towards this highly challenging tumor entity.
Fantastic. Well, thank you all for those great presentations. We're now going to start our Q&A session, so I invite you to join me here on camera. Also, a note to our audience to keep sending in your questions. Anything that we don't manage to get to today, we can reply over email. So our first question here. How do PDAC organ-on-chip models compare to PDAC animal models? And I wondered if we could come to you first here, Nicole.
Sorry, I needed to unmute myself. Okay, always the same mistake. Yes, okay, so animal models are, of course, very important in this research, and we cannot replace them entirely. But as Alina has already mentioned, most of the animal models are immune deficient, actually, and this is a problem when you study the effect of immune cells in particular. And the second thing is that animal models are focused on selected driver mutation, but the tumor itself is much more complex than just a simple RAS mutation. So there are several cell types which are playing a role. There are several also epigenetic changes which are playing a role, and this cannot be reflected by a mouse which is genetically engineered via a RAS mutation, for instance. And the third argument is actually a simple physical size argument. So a mouse is pretty small, and you can imagine, as a pancreas is very close to a liver in such an animal, you have a liver metastasis instantaneously. And this might not reflect the process which is relevant in a patient, such as, for instance, infiltration via lymph vessels or via neurons.
Great, thank you for that answer, Nicole. Now we've got another audience question in here, and it's a bit of a long one, so bear with me. Spheroids treated with specific monoclonal antibodies, TILs, along with inhibitors for Treg cells as combinatorial therapy for anti-tumor therapeutics for complex TME of solid tumors. In that context, how do these chips allow for maintaining better interacting environment for these therapies to the specific biomarkers? Tom, can we come to you here?
Sure. That's a wonderful question. I mean, we are really just getting into this to look in this in the PDAC context, really looking at T cells and how they look like. I mean, we already showed you here that macrophages really polarize in that direction. So, of course, now intervention therapy to target these tumor-associated macrophages is now possible in CHIP. Also, of course, we do have the endothelial barrier above, so every kind of intervention, antibody intervention, or drug intervention will have to go through an endothelial barrier and really cross this first before it will even go to the tumor. So also there, we have this additional complexity that it's not so easy to get in another or the normal in vitro setting. So there's multiple levels that we can look at here, and of course how T cells are going to polarize in there, we'll see. What I can tell you already is that just the stretch and strain stress that also the T cells have as we're perfusing them through the chip and through the tubing, that adds to kind of a baseline activation of T cells. And if we're then looking for example, antibody therapies and T cell, not necessarily now in PDAC where we haven't really done this, we can see that it really changes depending on whether you have them static or perfusing them. So there are differences, and chip in general will make it more in vivo-like than the 2D model.
Yeah, that makes sense. Thanks for that, Tom. And next question here: how do you keep your cells or spheroids healthy regarding temperature and CO2 concentration throughout your experimental runs? Alina, maybe coming to you for this one.
Yeah, of course. So the chips are actually located in the incubator together with the pumps. So we have the same temperature and CO2 concentration as we would have for normal 2D cultures. And actually, the chips allow us to create gradients, for example, for oxygen and nutrients that we also would have in the normal human. So replicating the situation we have in vivo more better, and yeah, so it's the same as for normal 2D culture. We cultivate the chips in the incubator.
Fantastic, thanks for that answer, Alina. And next question here: how long does it take to set up the chip? For example, how long do you need to culture the spheroids before applying an endothelial layer? And is it possible to preserve the chips, such as for shipment or to another lab or location, or do they need to be set up de novo each time? And then how long can co-culture continue?
Well, maybe I can get started. There's a lot of different questions in this. So first, we usually set up spheroids outside. So they are set up in ultra low adhesion plates, and then we bring them into the fully established chip where an endothelial layer is already there. So in that case, the endothelial layer takes about maybe 2 to 3 days to form, to have a tight layer that's perfusable. The spheroids are formed, and then 4 to 6 days after they've been formed, they will be put into the chip. So you can kind of time everything together to set it up. For the second part was shipping. I mean, generally it's like a tissue culture device. Shipping is not really advisable. I mean, certainly as soon as you get out of the incubator, the cells will get stress as they used to have. So you have to set it up at your own lab and really then do it there. I think there was another part, but I forgot about it.
The last part of that was how long can co-culture continue?
Yeah, maybe Alina, do you want to say something about this?
So normally we co-culture for 72 hours, but the spheroids are actually stable for a much longer time. So it should be possible to prolong the incubation time here.
Yeah. Absolutely. Maybe to add on this, is exactly what we do in the PDAC model. I have other tumor models that I run for 2 weeks, 3 weeks. It's really a little depending on the vasculature, how long that is stable. But tumor culture can go over a very long time also in chip.
So we have experiences with this model. It's already 4 years ago or so where we generated this experience, and actually the models are viable up to 6 or 7 days. This was the longest time point that we checked.
Great, thank you all for those answers. And unfortunately, that's all we've got time for today, but thank you very much, Nicole, Tom, and Alina, for answering those questions. And any audience questions that we didn't manage to get to, we can reply via email. The webinar will be available on demand tomorrow, so do look out for an email from us with the link. And we'd also like to invite you to take a short survey to provide us with feedback on today's webinar, the link to which can be found in the resources on the right-hand side of the screen or in the on-demand email. And an attendance certificate is available on the right-hand side too. And all that's left is to thank Nicole, Thomas, Alina once more for a great presentation today. And thank you to the audience for listening. We hope you'll join us again soon.